Hello, my name is Niall McKay and welcome to Living with PSC, a podcast about the rare disease of primary sclerosing cholangitis. Now, PSC causes the bile ducts inside and outside the liver to become scarred, narrowed and eventually blocked, and leads to liver cirrhosis. In this podcast, we aim to dispel some of the myths about the disease. Welcome to Living with PSC, and today we're talking to Jesse Kirkpatrick. And Jesse, do you want to tell us where you are at the moment?
Yes, so it's great to be here. Thank you for having me. I'm currently a third-year medical student at Harvard, and I'm also a researcher at the Massachusetts Institute of Technology, working in the lab of Dr. Sangeeta Bhatia. Having already completed my Ph.D. before starting med school, angling for a career as a physician scientist.
Oh, wow. And what was your Ph.D. in?
So my Ph.D. was in biomedical engineering, and the types of things I was working on are very relevant to the research that we'll be talking about today.
So you'll be doctor.
I'm already getting the business cards made up.
Yeah, you're already a doctor, but you'll be a medical doctor as well.
That is true.
So did you do your undergraduate in science as well?
I did, in biomedical engineering as well.
Are you like these medical shows you see where the med student is older than doctors?
But yes, it is definitely true that I am the oldest person in the room.
Yeah. Well, we're delighted you are, because we're delighted about the experience that you bring. And obviously the PhD in biomedical engineering didn't go to waste. Would you like to tell us about, well, first of all, congratulations on your award. Would you like to tell us a little bit about the award and what it's about?
It's the American Medical Association Research Challenge. It is a research competition that's open to all medical trainees, I think, in the US.
And would you like to tell us your connection with PSC or why you're interested in it?
I was exposed to PSC much earlier than other people who enter into the medical field because of a family member who has PSC, which is what inspired me to become interested in the world of medicine in general and in PSC in particular at the age of 17. It's
obviously heartbreaking for us when we get diagnosed with PSC, but you know, one has to feel for the younger people. It seems very, very unfair.
There is a bit of a misconception, even in the medical community among people who don't treat PSC patients, that PSC is a disease that is primarily like an older person's disease, and that's the way that it was taught to me in medical school, which of course made me cringe a little bit in my seat, knowing what I know about PSC and the diversity of people that it affects. And I think that's for a conversation for a different podcast about how do we, as a community, as a patient community, and a community of people who care about this disease, make people understand what PSC really is.
Tell us about your research is in cholangiocarcinoma, which is liver cancer of the bile ducts.
A cancer that arises in the bile ducts, although it can affect any part of the entire biliary tree, as we call it, from inside the liver all the way down to where the bile duct connects to the intestine.
Tell us what the disease is, I suppose, if you can, in as layman terms as you can manage.
So cholangiocarcinoma is a cancer of the bile ducts. One of the 500 functions of the liver is to produce bile. And bile is important for digesting fats. The bile molecule is kind of like soap. It can absorb fats in a way that water or other types of liquids couldn't.
It's acidic in nature.
Not necessarily. The acidity of the bile is actually regulated by the bile ducts. It does play an important role in how well the cells in the bile duct are actually able to handle and are able to handle like the conditions of the bile. The more acidic that the bile is, the more likely it is that the cells in the bile duct are to be damaged.
Yeah, and then of course that's the problem as well, isn't it? Because once the bile ducts become blocked, then the bile backs up into the liver and causes, liver cirrhosis.
Exactly. And there are some theories about, what causes PSC that have to do with the bile becoming too acidic, which causes the bile to cause injury to the bile duct cells, which then can cause, some of the narrowing of the duct and the blockages and all the things that are associated with PSC. Fundamentally, zooming out, the bile ducts are what transport the bile from the liver all the way to the intestine, where the fats that you consume in your diet mix together with the bile in a way that allows those fats to be absorbed and used by your body. And so one of the symptoms of a bile duct obstruction is that your stools become fatty. And that's because the fat in your the fat that you in your diet is not getting properly absorbed by the bile. So cholangiocarcinoma is quite a devastating and deadly cancer that affects the biliary tree, the bile ducts that connect, from the liver all the way to the small intestine. The 5-year survival rates are quite poor, around 10%. The survival rate is much better if you detect it early. And the reason for that is that if the tumor is detected early, it can be treated most often with surgery. In PSC patients, the most common surgery that's done for early stage bile duct cancer is actually a liver transplant, which was pioneered by the Mayo Clinic and can actually be a cure for bile duct cancer as long as it is detected at an early enough stage. But unfortunately, the vast majority of bile duct cancers in PSC patients are detected at an advanced stage that would not benefit from a surgery. And so that is kind of the problem that we and others are hoping to address by developing better diagnostic or screening tests for bile duct cancer.
And indeed, like PSC is often not detected until 5 or 10 years after, or longer in a lot of cases.
That's also true. We know from studies that have been done in Europe, there are many patients who have PSC but who don't know it. They have IBD that with changes associated with PSC that you can see by MRI imaging, but that aren't actually manifesting clinically. And so these patients wouldn't necessarily be diagnosed with PSC until, years down the line.
Yeah, I think in my own case, like I was diagnosed in 2015, but I do remember having really bad fevers and brain fog in around 2000.
I do think it is an important question. And I think there have been discussions around, better using like patient interviews to better understand what are these earlier presenting symptoms to better understand how PSC presents initially.
Anybody who does have any kind of liver disease will usually find a CA19 test on their blood form. And can you tell us what a CA19 test and also what you think the effectiveness is of detecting any type of bile duct cancer?
So CA19-9 is what we call a biomarker, which can give you information that helps guide clinical decisions around cancer. It is a molecule that is made by bile duct cells and is also made by cholangiocarcinoma or bile duct cancer cells. It is in patients who have bile duct cancer, that level is more likely to be elevated than in a healthy individual who doesn't have any type of cancer. But there are two measures that we think about, or two fundamental measures, which are sensitivity and specificity. Sensitivity basically tells you if you do have the disease, how good is the test at telling you that you have the disease? And specificity is if you do not have the disease, how good is the test at telling you that you do not have the disease? There are 10 to 20% of people who don't make it at all, and that even people who have other types of inflammation that are not related to cancer can have elevations in the CA19-9. Those tell you that the sensitivity and the specificity, respectively, are not ideal for that test. And so I think that there are many clinicians that would argue that it is still a useful test. And in the right context, the CA19-9 number can give them information that can help to guide clinical decisions. I think that there are also reasonable clinicians that would argue that CA19-9 isn't a test that's worth getting because it can give false hope to people who have negative numbers and false concern and worry to people who have positive numbers.
And this brings us nicely to the research that you're doing, because your aim really, I suppose, on a broader sense, is to be able to detect the cholangiocarcinoma more quickly, is it?
More quickly and more accurately.
Can you tell us a little bit about the method that you're using?
I think it'll be helpful just to mention, I think the most standard screening test that is used for people with PSC for cholangiocarcinoma, which is imaging tests, and in particular MRI and MRCP. And I think that MRI, MRCP it's proven itself to have reasonably good sensitivity. So it's able to pick up small bile duct tumors. But I think that where it falls short is in specificity. It is very difficult to distinguish early stage cholangiocarcinoma from the typical, narrowing and obstruction and inflammation and fibrosis that is associated with PSC, even in the absence of cancer.
I'm sorry, I interrupted you there. You were just getting to the nub.
What we asked was, can we develop some kind of a contrast agent that could help clinicians distinguish those malignant or cancerous lesions of the bile duct from benign or normal. There has been this new type of diagnostic technology that has emerged over the last few years, which are called activatable diagnostics. And so the way that activatable diagnostics work is that you can actually deliver something into the body that is able to accumulate at the site of a disease and then release some kind of a signal. So it can be activated at the site of disease to release some kind of a signal to the outside world that there is a disease process going on here. And the specific signal, the activation signal, if you will, is a certain type of enzyme that cancer cells use to invade, to spread to other parts of the body, to recruit blood vessels. And those enzymes are called proteases. So pretty much every single cancer type you can think of uses proteases as, to do what cancers like to do, which is all those things that I mentioned. And so what our lab has done is we've developed a method to detect those proteases, which are made by bile duct cancer cells, but not fibrotic or scar tissue cells, as a way to distinguish patients who have cancer from patients who have benign diseases like fibrosis. And I'll just say right off the bat that we haven't actually tested this in patients yet. We've only tested this approach in a mouse model of bile duct cancer and a mouse model of bile duct fibrosis. But this is the first step in further testing. The
device that you use, or the particle that you use to gather that information, is it a nanochemical compound?
It is a peptide, and peptides are basically short protein fragments. And the way this particular probe works is there are three components to the peptide. There's a positively charged part of the peptide, a negatively charged part, and then those two pieces are connected by a piece that can actually get cut by the protease. I didn't mention this before, but what proteases do is they cut proteins. And so what we're doing is we're delivering kind of a synthetic protein that is specifically designed to detect the specific proteases that are made by the bile duct cancer cells. And because there's this positively charged and negatively charged part, those two pieces kind of stick together. They, positive and negative attract, both chemically and I guess maybe in the real world, opposites attract. And so when that before, like in its intact form, the positive and negative pieces are stuck together. But when you deliver this piece, when you deliver this probe into a patient, for example, who has cholangiocarcinoma, and it accumulates in the tumor, the proteases cut that piece that's connecting the positive and negative parts. And when that happens, they disconnect. And the positively charged part is actually able to stick to the cells in the tumor. This is a property that's well established over many, many years, that positively charged peptides, or these short proteins, are able to bind to and actually be internalized by cells. And so these are actually called cell-penetrating peptides. And if you take some kind of a tag, like a fluorescent molecule or something that can be seen by imaging, and you attach it to that positively charged part, then you're actually those cells are also going to be bound by something that can be detected by imaging, like the fluorescent molecule. And so you're delivering all of this, these activatable particles to the tumor. The tumor is activating them, causing this sticky piece to stick to the tumor and light it up.
Got you. Yeah, no, it's very, very clever. The detection bit, you might have to change that slightly, will you, in terms of how you light it up?
Exactly. And so in mice, fluorescence is kind of an okay method, but fluorescence is kind of a sort of a fancy word for light. And if you think about trying to shine a flashlight through like the skin of a mouse, I guess not a lot of people work with, have a lot of experience shining flashlights through the skin of a mouse the way that maybe I do, but but you can kind of see through it. But when it comes to a person, if you stick a flashlight on your back and you try to see it through your belly, it's not going to work. Humans are a lot bigger than mice. And so what we're really hoping to do is to, instead of using fluorescent imaging, we're hoping to adapt this to a different type of imaging called PET imaging. And PET scans are routinely done for trying to assess, whether someone has cancer, where the if and where the cancer might have spread to other parts of the body. And so what we would want to do is kind of adapt our probe to be compatible with PET imaging, so that we could kind of get the benefits of being able to see to see where the tumor is and whether or not it's a cancerous cancerous lesion in the body.
Now, is there any treatment function with the molecule sticking to the cancer? Can it do anything about the cancer itself, or can it just tell you that the cancer is there?
So in its current form, it can only tell you that there is cancer there. However, we are at MIT are trained to think as engineers, and we always are looking for the next thing, and we're always thinking about how could this be adapted for other applications. And I think that what is really nice about this approach is that it's extremely quote unquote modular, and by that I mean there are multiple pieces that can be swapped in and out to exert different functions. And so you could imagine, instead of having a molecule that lights up the tumor and tells you where it is, you could instead have a molecule, for example, like a chemotherapy or some other type of molecule that has anti-cancer properties, and you could use it as a method to deliver those molecules to the cancer.
Yeah, because drug delivery for cancer is the sort of the golden fleece in a way, isn't it? Because the tricky bit with cancer is getting the chemotherapy not to kill the patient as well as the cancer. I mean, it'll kill the cancer, but it'll often kill the patient in the process.
No, exactly. And I think that targeted therapies are certainly, as you said, holy grail for in oncology. One approach that has come about in recent years is what are called antibody drug conjugates, which is kind of a similar idea. These are molecules that can be can also be administered into a patient. And because it has an antibody, these antibodies are designed to home specifically to molecules that are expressed by cancer cells. And so the antibody basically binds to those cancer cells, and then they get internalized, bringing with it the cargo that the antibody is holding onto, which is an anti-cancer drug.
It is fantastic work, and we really appreciate you doing it. And it's great as well. There's a link between Harvard Medical School and MIT, is there?
There is. The program that I'm a part of is it's called the Harvard MIT Health Sciences and Technology Program, and it's really designed for either future physicians who want to become skilled with the physical sciences, engineering, and research, as well as future scientists and engineers who want to have a closer connection and understanding of clinical medicine. And so the Ph.D. that I completed was actually also through the Health Sciences and Technology program. And I had the chance to do some actual clinical rotations, kind of play the role of a third year med student when I was just doing my PhD. And that was something that really inspired me to want to make that pivot and become a physician scientist myself.
How did you come to this idea?
I'm in the lab of Dr. Sangeeta Bhatia, who's also an MD PhD, who also completed both her PhD and MD through the very same Harvard MIT Health Sciences and Technology program. When I joined the lab, I was, of course, already interested in PSC and in cholangiocarcinoma. And I knew that Dr. Bhatia her lab had developed a similar but different technology that is also designed to detect proteases in cancer. And my goal from the very beginning was, how could I take some of these technologies that the lab has already developed and apply them to cholangiocarcinoma? And I would say that initially it was challenging. I think Sangeeta was very supportive, knowing my personal interest. But because of first of all, there was no mouse model that existed that actually modeled PSC-associated cholangiocarcinoma at the time, which in my line of work makes it extremely difficult to do any kind of testing. And then there was also kind of a paucity of funding for this type of work. And so initially, I shifted my attention to developing technologies for other types of cancer that we were funded to work on, and that there were mouse models, and those were prostate cancer and lung cancer. And so I worked on developing some of these technologies in those types of cancer. And once I completed my PhD, Sangeeta was kind enough to allow me to stay on for 1 year of postdoc, and then throughout my med school years so far, to apply these tools to cholangiocarcinoma. Initially, it was like kind of an unfunded project, and I was just doing it on the side. But eventually, we were able to get funding from the Resnick Center at Brigham and Women's Hospital to do this work in earnest.
You mentioned the Resnick Center. We're all very grateful to them and the Resnick Family Foundation, because they're funding all sorts of research in PSC, aren't they? And this is a very good example of how when you fund one area, a bunch of other areas tend to open up.
I completely agree with that. And I, of course, really echo your gratitude and appreciation to the Resnicks for this, their unprecedented generosity and commitment to PSC. And then I think also Josh Korzenek, who is, the head of the Resnick Center at Brigham, has been just an incredible steward of the center, and has done an amazing job of bringing together, as you mentioned, all these disparate areas that are relevant to PSC, but that might not have been directing their attention specifically on PSC in the past. And so I think that's one of the really great outcomes of that funding, is that folks who weren't necessarily devoting their technologies and skills and knowledge to problems specific to PSC are now able to do so.
And of course, you're on the board of PSC Partners as well, who are doing similar things in terms of funding research, but also drawing a lot of people together and a lot of disparate types of research together, which is, I presume, is part of your job on the board to read some of these proposals.
Yes, absolutely. And I think that PSC Partners really has done just such an incredible job of identifying promising research and just allowing that research to move forward in a way that wouldn't be possible without the funding that they've provided and the resources they've provided over the last many years.
What's the compound that you developed called, by the way?
So the general class, we call them activatable zymography probes, AZPs for short.
AZPs. So the AZPs that you have developed, would there be other functions that could they perform other functions within the bile ducts?
PSC, there's all kinds of proteases that are being expressed because proteases are also extremely important in the development of fibrosis, in inflammation. And so certainly I think that there could be, and actually maybe one thing I'll mention is that the first paper from our lab that ever described these protease activated nanoparticle diagnostics was actually done in collaboration with Yuri Popov at Beth Israel, using this MDR2 knockout mouse model of bile duct fibrosis. And so they were different types of nanoparticles. Of course, these were designed to detect the proteases that are made in fibrosis, whereas we're designing ours to detect the proteases that are made in bile duct cancer. But it just goes to show you that it is certainly that PSC and bile duct fibrosis in general are certainly a relevant application, both for diagnostic and potentially for therapeutic applications.
So it's kind of an exciting time in this area, isn't it?
And I will just mention, just since we're on the topic, that our lab and others are also actively working on developing a different type of technology that could be useful for PSC, which is these bile ducts on a chip, and other kind of ways to model what's going on in PSC using cells in a dish. And so we're working with certain types of cell type constructs called organoids, as well as bile duct on a chip type constructs. And those could be useful both in understanding what's causing PSC, as well as potentially testing therapies in a way that could be done more quickly and without immediately having to do clinical trials.
Organoids are really artificially grown bits of organs, really. Is that correct?
Exactly. Yeah. Thanks for clarifying that. Exactly.
At a dinner party, you can really, when somebody asks you what you do, you're like, well, I'm attempting to cure cancer.
I never really know what to say at the dinner party. I'm usually like, I'm just a lowly third-year med student.
Well, that certainly isn't the case. Well, is there anything that I haven't asked that I should have asked?
I just want to also acknowledge the other efforts that are out there to early detection of cholangiocarcinoma, specifically in patients with PSC. And there have been some, I think, really promising research studies that have been using more genetic methods to detect cholangiocarcinoma in PSC. There was one paper that came out that showed that if you take a bile sample that's procured during ERCP from somebody who has a suspected bile duct cancer, a PSC patient with suspected bile duct cancer, it actually does a pretty good job at detecting. If you sequence the genetic material, the DNA in that bile, it does a pretty good job of distinguishing the folks who actually have bile duct cancer from the ones who don't. Wow. And similarly, you can take there was another paper that showed that if you take a brushing of the bile duct, and instead of just looking at it under the microscope as is typically done, you sequence the genes using a targeted panel of looking for a specific genes that are associated with bile duct cancer. That actually also does a pretty good job at distinguishing cancer from benign PSC. So I do think that, and then there's another group that's led by Dr. Jesus Benalis, that's had a lot of success in using what are called extracellular vesicles, and these are kind of free floating organic, sort of nanoparticles that your own body makes, and those can be and actually measuring certain molecules that are produced in those extracellular vesicles has also been shown to be pretty effective in distinguishing bile duct cancer from benign PSC.
Is there any indication or risk factors for, that's creating the bile duct cancer as opposed to, people who don't get it?
It's a very good question, and honestly, one that I'm very interested in, and one that really I think has a lot of opportunity to investigate further and is not well understood. You could kind of come up with hypotheses about why PSC patients are so predisposed to bile duct cancer. Obviously, there's a lot of inflammation that's going on. We know from other types of cancer that inflammation certainly can play a role in causing tumors to form. Fibrosis itself it can change the stiffness, for example, of the liver. They could make it more likely for cancer to form. Or it could or potentially there are genetic risk factors in PSC patients that also predispose to bile duct cancer. There have been some studies looking at the microbiome, the bacteria in the gut, and the role that could play in the development of bile duct cancer. And of course, we know that in PSC patients, the bacteria in the gut are different from in people without PSC. So I think there are all kinds of hypotheses. There at this point, I'd say there are more questions than there are answers. But I think it would be an area that would be absolutely worth investing in.
Dr. Jesse Kirkpatrick, thank you very much for your help today. It's been a pleasure having you on the podcast.
Thanks for having me.
Well, I'm afraid that's all the time we have for this week. For more information about PSC and for a list of people and topics we'll be covering in the near future, please go to www.pscpartners.org. That's www.pscpartners.org. PSC Partners is a non-profit organisation dedicated to providing education and support to primary sclerosing cholangitis patients. There is information and resources on the website. There is a conference once a year. There is a private Facebook group that you can also join. Also, please join our patient registry. This is an anonymous database that provides research support to the medical community who are researching this disease. Thank you very much for listening.